Well, hey guys, welcome back. This is our third section of notes. This is over the neuroglia. If you recall, we introduced this section of notes at the end of the neuron notes to kind of give you a little bit of a heads up as to what this is all about. So get out your notebook, write this information down in the input section of your notebook. That would be the right-hand side. And let's get started. Here's your essential question. Your essential question today is to compare and contrast the oligodendrocytes and the Schwann cells using a Venn diagram. So you can start your comparison with these two images here. Here's the Schwann cell wrapping around what we can see as a cross-section of an axon, and we have the oligodendrocytes right here located next to a neuron. All right, as we go through these notes, you will be able to answer this question. And also, don't forget, this is in Cornell note fashion, so you need to make sure you summarize and add some more questions for possible test questions so you can study really well for your test. All right, so these are the neuroglial cells that are found in the central nervous system, okay? So that would be your brain and spinal cord, their central nervous system. We've got the astrocytes. Astro, like astronomy, they're star-shaped, okay? They're star-shaped cells. They form a blood-brain barrier by covering blood capillaries. Okay, so your brain itself doesn't have blood in it, but astrocytes cover the blood vessels up so blood doesn't leak into your brain. If blood does leak into your brain, it often ends up in a very, very bad situation called an aneurysm. You don't want that. What does an astrocyte do? Well, astrocytes metabolize neurotransmitters. They also regulate the balance of potassium ion. In our next section of notes, we're going to be talking about potassium ion and sodium ion and the role that they play in an action potential. And astrocytes also provide structural support of your central nervous system. Now, the second type of new neuroglial cell we're going to discuss is the oligodendrocyte. Oligodendrocytes are the most common glial cell type, okay? Most of your glial cells, your neuroglial cells, are oligodendrocytes. And what these do is they form a myelin sheath around more than one of the axons in the central nervous system. And they're similar or analogous to the Schwann cell of the peripheral nervous system. Essential question. All right, next two types of neuroglial cells, we have the microglia. Microglial cells, we can see here, they're small cells that are found near the blood vessels, and they play this phagocytic role, or phagocytic, however you want to say it. What that means is they will consume and clear away dead cells and debris. So these microglial cells are really useful cells. They're our little trash compactors. They come in and they clean up the mess that maybe something didn't work right, and it comes in and cleans it all up the mess. All right, also from like bacteria or viruses and stuff too. Microglial cells are really, really working hard. These microglial cells are derived from the cells, those stem cells, that also gave rise to macrophages and monocytes. Macrophages and monocytes are white blood cells that we have. Ependymal cells. Ependymal cells form an epithelial membrane lining of the cerebral cavities in the central canal that surrounds the spinal cord. Now, these guys are responsible for producing cerebral spinal fluid. So, we said that there's a blood-brain barrier, so how does the brain stay wet? How does the spinal cord stay wet? So that all of these fluids and all of these different proteins and stuff like that can move back and forth. It's through the cerebral spinal fluid that this happens. Okay, now we have two types of cells that are neuroglial cells in the peripheral nervous system. Satellite cells, which are flat cells surrounding neuronal cell bodies in the peripheral ganglia, and they also support neurons in the ganglia. So these satellite cells are going to be found in the peripheral nervous system near the ganglia. The ganglia are those big branches of bundles of nerves that branch out from the spinal cord. Also, we have the Schwann cells. Essential question. Schwann cells are cells that move, or they encircle the axons of the peripheral nervous system. Each cell produces part of the myelin sheath surrounding an axon of the peripheral nervous system. All right, so you can see here that these are the neuroglial cells. There are 6 total neuroglial cells that you need to know for your future. Now, myelin. We've mentioned myelin on a couple of occasions, both in the central nervous system and the peripheral nervous system. The oligodendrocyte is the cell that's responsible for it in the central nervous system, and the Schwann cell in the peripheral nervous system. Now what this does, what myelin is, it's an insulating outgrowth of these cells made of lipids and proteins as an extension of their membranes. And what they do is they provide insulation almost to speed up an action potential. They make it so that the action potential can zoom across that membrane very, very quickly. And what it does is it wraps around the axon of the cells, and whenever an embryo or fetus is developing, myelination begins at the 14th week of development. Now, a little bit of myelin exists in the brain at birth, but then during infancy, as the child continues to grow and develop, myelination occurs quickly. That's why babies, they don't seem to react very quickly. It's because they don't have a whole lot of myelin in their brain. But during adolescence, your teenage years, like you guys are, teenage years, you got really, really quick reflexes, okay? It's because your myelination is at its peak when you're a teenager. Now, gray and white matter, okay? The difference between gray and white matter in the central nervous system or in the peripheral nervous system as a whole. White matter is the matter that's myelinated. It's white in color, all right? Gray matter are the nerve cell bodies, which includes the dendrites, the axon terminals, and the bundles of unmyelinated axons in neuroglia, okay? So white matter in the nervous system means it's myelinated. Gray matter means that it's not myelinated, okay? In the spinal cord, your gray matter forms an H-shaped inner core surrounded by white matter. And in your brain, there is a thin outer shell of gray matter that covers the surface and is found in clusters called nuclei inside the central nervous system. Now, your myelinated central nervous system. Remember, the oligodendrocytes myelinate the axons of the CNS. What they are is they're broad cell, flat cell processes. They wrap around the central nervous system axons, but the cell bodies do not surround the axons, okay? So there's no neurolemma that's formed, and there's little regrowth after injury due to the lack of a distinct tube or a neurolemma. Now, for the peripheral nervous system, the neurolemma is the cytoplasm in the nucleus of the Schwann cell. Cytoplasm in the nucleus. Notice how the Schwann cell wraps around the axon. Okay, so it's a little bit different. The Schwann cells wrap around the axons during fetal development. So, during the development before a child is born, this is happening in their peripheral nervous system. The Schwann cell cytoplasm in the nucleus forms the outermost layer of the neurolemma, with the inner portion being in the myelin sheath. Now, for unmyelinated fibers, they're slow, they have small diameter, keyword slow. The action potential does not flow through them as fast as it would with a myelinated fiber. Now, they're only surrounded by the neurolemma, but there is no myelin sheath wrapping, okay? So in the peripheral nervous system, it is possible to have a neurolemma around the axon and there be no myelin sheath. All right? All right, so that concludes the notes for today. Make sure you go back and answer that essential question, your comparison and contrast between the oligodendrocytes and the Schwann cells. Give a lot of detail in that response, because you're going to need to know that in your future. Next time, guys, we're going to talk about action potentials and all of the details involved with that. Have a great day.